In this module, we are going to see about HDL metabolism, which is high density lipoprotein. Okay, HDL is high density lipoprotein. Now, this is something important to you all. You should definitely learn this topic, right? So we'll see from where is this HDL synthesized. From where is this HDL synthesized? It is synthesized from the liver and also from the intestine. Okay, it is synthesized from the liver and also from your intestine. So from both these areas, how is it synthesized? It is synthesized as a nascent HDL. Okay, nascent HDL. Why do you call it nascent HDL? Because the shape of this HDL is discoidal. The shape of this HDL is discoidal. So why is it discoidal in shape? Because it has predominant phospholipid content, which is mostly lecithin. Okay, so phospholipid content. So it forms a bilayer. Okay, bilayer. So this is how the HDL molecule is synthesized. So on this HDL will be the apoproteins, ApoA, ApoC2 and ApoE. So what are the apoproteins present? ApoA, ApoC2 and ApoE. Now what happens? This is nascent HDL, right? So we have to make it mature. So before that, what do you know about this ApoC2 and ApoE? What have we learnt before? In the chylomicron metabolism or the VLDL metabolism, we've seen the nascent structures are converted to mature forms as they occur C2 and E from the HDL. Yes, as they occur C2 and E from the HDL. Do you remember? So this happens routinely in the circulation. So after the metabolism is over, the C2 is given back to the HDL in the circulation. So this is the function of the C2 and E. So HDL molecule serves as a reservoir of C2 and E so that it converts the nascent forms of chylomicron and VLDL to its mature form. Now, what is the function of HDL? The function of HDL is to transport cholesterol from the peripheral tissues. Yes, from the peripheral tissues to the liver. So, what is our idea? Our idea is to collect the cholesterol from all of the peripheral tissues. So, in cell, okay, within the cell there is cholesterol. So, how is this cholesterol given to the HDL molecules? By a protein called cholesterol efflux regulatory protein, cholesterol efflux regulatory protein, which is an ABC protein, which is a ABC cassette proteins. Okay, so this will efflux the cholesterol from within the cell or the cell membrane to the HDL molecule. To the HDL molecule. Now, this cholesterol which is effluxed from the cells gets into the HDL molecule. This cholesterol, if it is left free, it can easily get out of this HDL molecule. So we have to trap it inside this HDL by converting it into a cholesterol ester. By converting it into a cholesterol ester. Now this is done by an enzyme called LCAT. What is this LCAT? LCAT is nothing but lecithin cholesterol acyl transferase. LCAT is lecithin cholesterol acyl transferase. Now, what is the function of this LCAT? Okay, LCAT is lecithin cholesterol acyl transferase. I've already told you nascent HDL has more of phospholipid content, which is mainly lecithin. So, lecithin cholesterol acyl transferase. So, what does it do? The ApoA1, okay, ApoA1 which is present on the HDL molecule activates this LCAT, okay, activates this LCAT. So this LCAT converts lecithin, yes, lecithin to its by combining with cholesterol. So lecithin to combining with cholesterol forming cholesterol ester and lysolecithin. Cholesterol ester and lysolecithin. So this lecithin cholesterol acyl transferase, what does it do? It converts the cholesterol to cholesterol ester by simultaneously converting this lecithin to lysolecithin. Okay, so ApoA1 here activates the HDL. ApoA1 in the HDL molecule activates the LCAT, right? So now what happens here? The cholesterol is trapped inside as cholesterol ester inside the HDL molecules. Now this HDL molecule will now gain more of cholesterol ester now, right? So it will have more of other lipid content. So now it becomes spherical. Yes, now it becomes spherical as it acquires more of cholesterol from all the tissues. Now it becomes spherical. So this spherical mature HDL will be called as the HDL3. Okay, HDL3 molecule. So now what happens again, the apoproteins present here will be the ApoA1, C2 and E. So what are they? ApoA1, C2 and E. Right? So these are the apoproteins present on the mature spherical HDL3. Now what happens in this? Now it is in the circulation now. So it will acquire more of cholesterol esters inside the core. So as it gets more of cholesterol from the other cells in the peripheral tissues. Now what happens as it is in the circulation, now we've got our VLDL molecules also, right? So VLDL molecules will have what? VLDL, LDL molecules will have what? They will have cholesterol ester and triglycerides in them. Triglycerides in them. So there is more of triglycerides in the VLDL molecules. Now this VLDL molecules will give off its triglycerides to the HDL. Okay, it will give off its triglycerides to the HDL. Okay, it will give off its triglycerides to the HDL and in turn get this cholesterol ester from the HDL. Okay, in turn get the cholesterol ester from the HDL. So this is happening with the help of cholesterol ester transfer protein. Cholesterol ester transfer protein. Cholesterol ester transfer protein transfers cholesterol ester from the HDL to the VLDL molecules and also from the triglycerides from the VLDL to the HDL molecules. Okay, so not just VLDL, also from IDL, LDL molecules present in the circulation. So this is done by cholesterol ester transfer protein. Okay, so now this HDL molecule which gets more of triglyceride content in them from the VLDL, IDL, LDL molecule will be called as the HDL2 molecule. Okay, HDL2, right? So this is again the HDL2. Now, so the HDL3 can become HDL2 with the help of cholesterol ester transfer protein. So this is due to increased circulating LDL molecules, VLDL and IDL molecules in the circulation. Now, what is the fate of this HDL3 and HDL2? This HDL3 and HDL2 can again go to the liver because our ultimate function of this HDL will be to transport cholesterol from the peripheral tissues to the liver. So as it has transported, as it has got more of cholesterol from the cells, it will go to the liver. Yes, it will go to the liver finally. So in liver, what happens? There is a receptor, which is the protein called as the scavenger receptor B1 protein. Scavenger receptor B1 protein. Here what happens, this scavenger receptor B1 protein gets all the lipid content from the HDL molecule and it gives it to the liver. Okay, it will take off all the lipid content from the HDL molecule and it give off into the liver. So the HDL molecule again will now be released into the circulation as a nascent HDL. Okay, so this mature HDL which contains more of cholesterol ester in them goes to the liver wherein the scavenger receptor B1 protein will take off the lipid contents and on the lipid contents are taken up by inside the liver. Now this lipid contents will now be utilized further for bile acid synthesis. So that's how the cholesterol can be excreted by bile acid synthesis. Okay. Now also if it is HDL2 molecule, the HDL2 molecule we know there is triacylglycerol content in them. So what can we do with this triacylglycerol? There is one more enzyme called the hepatic lipase in the surface of the liver. This hepatic lipase can break down all these triacylglycerol, after which the scavenger receptor B1 protein can take up all the lipid content and it will give off into the liver. It is taken up by the liver through the scavenger receptor B1 protein. All the triglyceride content of the HDL2 molecule will now be broken down by the hepatic lipase, after which the lipid contents of this HDL molecule can be taken up inside the liver. So this is what happens, which is the fate of this HDL2 and HDL3. Now, what are the important proteins which you have to remember? One is the cholesterol cholesterol efflux regulatory protein, which is a ABC cassette protein, and then lecithin cholesterol acyl transferase, and then cholesterol ester transfer protein, and then scavenger receptor B1 protein and hepatic lipase. Okay, so these things you have to remember. These are the important points which you should not miss whenever you are asked such a question, right? So we'll see again. The first one is it is synthesized, nascent HDL is synthesized from the liver or the intestine. Okay, so from in it is nascent or it is because it is discoidal in shape, and it is discoidal because it contains phospholipid bilayer with more of lecithin content in them. The apoproteins present are the ApoA, C2 and E. Now this nascent HDL, as it passes through the peripheral tissues, from the peripheral tissues, from inside the cell, the cholesterol is influxed out of the cell and is taken up by the HDL. Now, so to trap these cholesterol, we are converting it into cholesterol ester with the help of lecithin cholesterol acyl transferase. Okay, now thus as more of cholesterol becomes cholesterol ester, it becomes spherical, mature, and this is now called the HDL3. Okay, now it is called as the HDL3. Now this HDL3 will have again ApoA1, C2 and E. Now this LCAT is stimulated by activated by ApoA1. Now this HDL3, as it goes through the circulation, it will encounter the VLDL, LDL and IDL molecules. Now here what happens, it will transfer the triglycerides from the VLDL, IDL, LDL molecules to the HDL in return for cholesterol ester from the HDL to the LDL, VLDL molecules. These through what? Cholesterol ester transfer protein. So the cholesterol ester transfer protein will transfer triglycerides from the VLDL to the HDL. So this HDL will now contain more of triglyceride content and cholesterol ester content will again be reduced. Okay. So this is now called the HDL2. Now this HDL2 and HDL3 has its fate in the liver. Now here there is a protein called scavenger receptor B1 protein, which takes up only the lipid contents from the HDL molecule, and the rest of the HDL will be again released into the circulation as nascent HDL. So which will again go through the circulation to peripheral tissues to collect the cholesterol. So the cholesterol which is taken up by the liver, it is used up for bile acid synthesis. Now again, if the triglyceride content is present in HDL, it will be broken down by hepatic lipase, after which all the lipid contents will be taken up by the scavenger receptor B1 protein on the liver. Okay, so the formation of this triglycerides in the liver, the transfer of these triglycerides from VLDL, LDL to the liver occurs only when your LDL levels are more. Okay, so it is actually not good. So the LDL levels have to, because we know that LDL is a bad cholesterol. Yes, again the cholesterol from collected from the tissues, we are again losing it here and we are taking up triglycerides here. So this you have to keep in mind. So why is this? What is the whole of this metabolism called? The whole of this metabolism is called reverse cholesterol. The whole of this metabolism is called reverse cholesterol transport. Yes, it is reverse cholesterol transport, because we are transporting cholesterol from the peripheral tissues to the liver. So this is called reverse cholesterol transport. And HDL, it is a good cholesterol because we are clearing off the cholesterol contents from the peripheral tissues, your blood vessels and everything, and you're giving it to the liver, where liver utilizes it or excretes the cholesterol by forming bile acids, right?